There we go. All right, it's going to take me a minute or so to introduce. So good afternoon, everyone, and thank you for joining this session number 3 in what is 4-part series of the virtual Podium Australia Pacific version for metabolomics and lipidomics. I think everybody who's attended the last couple of weeks would agree that we've had an extremely high caliber of talks and speakers from early career researchers all the way through to not so early career researchers. The format of today's meeting will be a series of lightning talks bookended by 2 keynote speakers. And the keynote speakers will be 15 minutes with 5 minutes available for Q&A, and the lightning talks will be 5 minutes each, running concurrently, and at the end of all of the lightning talks, we will have a combined Q&A session. If you look in your Zoom panel, you will find that there is a Q&A box where you can put your questions. So don't be shy, put them in there, and we will address them when we can. We are ready to go, and so I will ask you all to sit back, relax, and enjoy, and perhaps be inspired and collaborate. And I will hand off to Fana. And I just realized I didn't introduce myself. Hi, I'm Brad. But Fana's going to introduce our first keynote. Over to you, Fana.
Thank you, Brad. Welcome everyone. And it's really my great pleasure to introduce Professor Guanghe Shui from Chinese Academy of Sciences, Beijing in China. Professor Shui is going to present some really interesting data about COVID-19 lipidomics and metabolomics. So it's up to you. And now, Professor Shui.
Thank you for the introduction, and also actually thanks for the invitation. I will share my screen first. Everyone can see it? Can you see it?
Yes.
Yeah, great. I will start now. And today I'm going to talk about the omic driving system integration of the metabolic dysregulation of this COVID-19. Just one moment. Of the COVID-19 pathogenesis. First, I will talk a little bit about the metabolized infectious disease and metabolic disorders. As we know, actually, foreign metabolites are essential nutrients for our daily life, and adipose tissue is the main site for storing energy, and lipids such as phospholipids, sphingolipids, cholesterol are major components of cellular membrane, and also circulation metabolites are major components in our blood. Dysregulated lipid metabolism has been reported in various diseases, including cancer, cardiovascular disease, obesity, fatty liver disease, type 2 diabetes, as well as a virus infection. For instance, in virus replication, viruses are known to induce profound changes in host cell lipidomes and user K energy pathways in their exploitation of the host metabolic resources for fueling the different stages of virus infections. I gave one example regarding host pathogen interaction, which actually I was involved in still in 10 years before, around 10 years still in Marcus lab. For TAG, we know the triacylglycerides, which is required for survival of the Mycobacterium bovis BCG during hypoxic dormancy. You can see in the dormancy, TAG level is much higher than after its resuscitation. So basically, the resuscitation needs the energy TAG. If you apply the THL, which is the inhibitor of the TGL lipase, you can see you can inhibit TH with the increased amount of the THL. You can increase the TGL lipase activity. Thus, the TG consumption is inhibited. Thus, the cell growth is inhibited. Another example I want to show you that case. Like, for example, the TB strains, like the modern Beijing sublineage, accumulated a lot of TAG species compared to the old Asian strain as well as the standard strain H37RV. Further, this is confirmed by sequence found it has upregulated TAG synthesis like TGS2. Lipid metabolism is also closely associated with virus infection. First, distinct membrane curvature and precise lipid composition can determine the ease of viral entry and replication. And disease-associated changes in plasma or serum lipidom had been previously reported in a number of studies regarding to the viral infection. For example, increases in serum plasmalogen PE in Zika virus has been reported, and a reduction in plasma TAG and free fatty acid and increase in numerous plasmalogen PE and plasmalogen PC were shown in mice administered with respiratory syncytial virus. And for virus infection targeting the respiratory tract and the lungs, the consequential development of the ARDS may also alternately implicated in inflammatory process. For instance, PUFA containing PC, PE, and TAG species were observed to decrease in non-survivor of ARDS compared to survivors, and increase in serum CAT free fatty acid were also associated with the ARD developments. Bioactive derivatives of PUFAs, such as 13-HODE and 9-HODE, were identified as mediators that induce and resolve inflammation triggered by influenza infection, respectively. Now I'm going to talk about the high coverage lipidomic and metabolomic phase system evaluation of metabolic dysregulation in COVID-19. This is the most recent data that we show that the COVID-19 pandemic presents an unprecedented threat to global public health. According to Johns Hopkins data, currently the confirmed cases over 55 million, and the deaths caused by COVID is over 1.3 million. Infection disease actually are complexly multi-system diseases. Metabolism is a key regulator of the COVID-19 susceptibility and recovery and survival. Therefore, comprehensive understanding of the metabolic disorder triggered by viral infection can effectively maximize the survival rate. Plasma will be ideally material to invest body metabolism upon COVID infection. Plasma metabolism mainly contains 70% of lipids and 30% of the polar metabolites. So this study will cover both of them. The experimental design are very simple. Basically, we collect plasma sample from the recruited subject, and carry out lipid extraction and metabolic extraction, then inject the sample into the LCMS systems. First, before we started our data, we show some of the demography and laboratory finding of this recruited subject. Basically, these data are in good agreement with published literature on clinical characteristics of the COVID-19 in China. A few key parameters, like including the T cell counts. We found reduction in T lymphocyte count decrease actually with increasing severity, and similar to CD4 T cell count. Another system we found the indices of the systematic inflammation, including C-reactive protein, interleukin-6, ESR data, and serum ferritin and procalcitonin levels. These data exhibited progressive increase as disease severity increases. Another data I want to show the data is the apoproteins. As we can see, there is not much change for ApoB proteins. It displays a V shape. However, it displayed a trend for gradual decrease of the ApoA1 with disease severity increase. So coming to our omics data, basically we first carry out a high coverage target lipidomic data analysis. This one you need to choose right separation method, ionization sources, and MS parameters. Basically, this part it was carried out using the Science 6500 plus the co-trap LCMS in combination with normal phase LC and reverse phase LC. And the target lipid species, which is actually similar to our last year's publication in WCares, target around 800 lipid species, including various phospholipids, sphingolipids, free fatty acids, isoglycerides, and sterols. Basically, they are quantified with 29 spike internal standards, which cover lipid species cover different lengths of fatty acid and different degree of unsaturation. For non-target metabolome work approaches, basically it's still using the Science Co-12, triple 12, 5600 plus. Basically, the coordination use the ideal MS model and the full-scale MS models. We target the metabolites we analyze, including isocarotene, amino acid, carbohydrate, and so on. The unique of the metabolomic study in that one is we spiked with different from traditional ones. We spiked with 45 labeled internal standard from different group of metabolites. This is trying to increase the analytical precision. So basically, the rules how analyze these rules according to 3 steps. First, the internal standard were applied to correct peak error of the corresponding metabolites. If the internal standard were not feasible, then due to a peak error will be corrected with internal standard metabolites of the same class or comparable peak intensity and or proximity in retention times. And the results from step 2 were evaluated based on the RSD value of each metabolite before and after ice correction. Correct peak error were adopted if their corresponding RSD values were smaller than that of original case in quality control samples. This measure actually currently under preparation. So after we get the lipidomic and metabolomic data, we will clean up the data. Basically, ratios of only ratios peaks with ratios of singular to noise larger than 4 will be kept. And the RSD value of the metabolite or COV of the metabolite from the QC sample will be applied to remove those peaks with big variations. So then we shortlist around 1,002 identified metabolites, including around 600 lipids and 400 polymerides to investigate the potential lipids or metabolites associated with disease severity. So how to select the biomarker candidate? First, we're starting from the variable with all p-value less than 0.05 between health control and patient after adjustment for age, sex, and by BMI, ranked by ascending p-values. Next, we do the variable elimination. We select a starting variable with lowest p-value from the starting pool, and then eliminate all remaining variables with significant correlations to the selected variable from consideration. Then start a second run. Then we repeat step 2 and 3 until all are exhausted. Then we establish the seeds. Basically, finally we have the 10 selected variable produced in part B, and establish 10 seeds by putting in variables with significant correlation p-value to select variable ranked by ascending p-values. Then each seed, top 5 variables in each seed will be presented. This including, as you can see, 10 seeds I'm showing here. And then we select the representative variable from each seed by lowest p-value and or report biological functions through PubMed search. And with this 10 select candidates, we build up the AUC, which is around 9.7 for predict disease And as you can see from the change of the concentration among this, most of this top 10, this 10 metabolite, most of them are lipids. Actually, for example, S1P and sphingomyelin show like different trends. S1P decreased drastically and sphingomyelin increased a lot, same to GM3 species. Next, we carry out a noise regression model. Basically, the noise regression model with covariate BMIH and 6 were built with each metabolite to search for significant variables that could predict disease severity of the subjects. First, let's look at the plasma metabolome. Several isocarnitines such as palmitocarnitin, sterocarnitin, and oleocarnitin were reduced in COVD. This indicating beta oxidation pathway were affected. And this basically reduced circulation level isocarnitine indicated annotation of intraval fatty acid into mitochondria for beta oxidation. Next, you can see we found the progressive reduction of the sulfate steroids were also observed, increasing disease severity, as well as many amino acids, including tryptophan, valine, and so on, were significantly reduced in mild and moderate patients. And more interestingly, we found metabolites constituting the TCA cycle were generally reduced in patient. The reduction in polar metabolites participating in oxidative pathway of energy production, including beta oxidation, I mentioned in previous slides, TCA cycle, particularly in severe patients, may indicate metabolic responses to decline non-functions and limiting blood oxygen to lower reliance on oxygen for cellular energy production. Interestingly, we also found ethylcholic acid, which is a macrophage-specific metabolite, were progressively reduced until the moderate. Ethylcholic level actual papers was reported to be positive correlated with the expression of the immune response gene 1 in both human and mouse immune cells. Next, we come to the lipidomic data. We found that reduction in major phospholipid species, including PC, PI, and PAs, and with a accompanying increase in their nisophospholipid species, which indicates enhanced phospholipase A2 activity in patient. Previously, it has been reported that cytosolic phospholipase A2 alpha activation was reported to trigger pulmonary inflammation following pathogen interaction. Therefore, change in phospholipids suggests reduction in circulation. This is which I showed the ApoA1 data as before as the COVID-19 progress. And interestingly, we also found for neutral lipids, there's a marked reduction in many actually DAG species with increase of concomitant increase of the free fatty acid in the both mild and moderate groups, with TAG were significantly reduced in mild cases. Previously, it has been reported that the increase in C18 free fatty acid diminished TAG species were in agreement with the previous the ARDS. Most interestingly, we found sphingolipids species like sphingomyelin and GM3. They displayed progressively increase with increasing severity, as you can see from the data. This possibly reflecting the augmented secretion of this lipid into the circulation. In line with this observation, our observations, previously report show that HIV membrane enriched in sphingomyelin and viral infection was upon time is over. Okay, quickly go through.
30 or 40 seconds, and then we'll start some questions.
Also, the sphingomyelin, that's a gross overview of the plasma sphingolipids as associated with independent baseline. This indicates a close resemblance to that of exosomal lipid composition, particularly enriched in sphingomyelin GM3 species. So the correlation primarily indicating like the phospholipids, basically they are correlation, they are negative correlation with inflammation in the indices. And only GM3 are negatively correlated with the T cell count and CD4 T cell count. So lastly, we carry out this network analysis, which indicate many lipid species, actually their correlation are lost in the disease. So I don't go through too much detail for this last two slides. For our summary, basically we found a comprehensive quantum lipidomic for profiling of COVID plasma, and we found that actually they provide a good biomarker, and there are different correlation analysis uncovered metabolic dysregulation in COVID-19. And we found GM3 sphingomyelin enriched exosome are positively correlated with the COVID-19 disease pathogenesis. Last slide for the future slides. Actually, we found we have a few patients actually during their discharge. We found some lipids they can return back, but many lipid species or metabolite they didn't return to normal. So this gives us some hints what happened to the future. First, I will stop my presentation and leave to questions. Thank you all. Sorry, I talk a bit more because initially I prepared for 20 minutes talk. Any questions?
Thank you very much, Professor. It's very interesting talk. I have one question regarding, you mentioned about the BMI you consider. Have you observed any trend between high BMI with the trend to the disease or COVID-19?
That's a very good question. Actually, BMI has been reported, to associate with disease in quite a few papers. So in our study, we didn't correlate with the BMI with the disease severity, because somehow actually we should do that, but we didn't do that. But there are already quite a few papers.
Okay, thank you.
Thank you, Professor. Sorry, thank you, Professor Shui. We have one question from the audience, Sneeza. She's asking, was there a comparison done of metabolites and lipid profiles between symptomatic and asymptomatic COVID-19 positive subjects?
We didn't do that. That's a very good point. Actually, maybe I can talk a little bit about this. Actually, we have one paper. We submitted one paper, this is one of the very small part of this. We have covered this. We address this question a little bit, but it's not the essential of our paper. Our paper currently is still actually under review.
Is there no question? I'm happy to ask one more question. You mentioned about tag 61, which is the 18-1, which is, have we seen the other lipid species related to 18 comprehensive data with that, or is there any connection between other lipid classes with the 18?
Sorry, you're talking about 16-1, 18-1 in which class?
Tags, triacylglycerols. In one of your slides you mentioned, and quite interesting.
This slide?
Yes. No, I think one before.
No, we triglycerides. We only this class.
Yeah.
This slide.
But do you see all the other lipids classes with the 18-1 are either decrease when tags increase, or is there any pattern between the lipid classes?
Oh, you're talking about maybe they're talking about more about the fatty acid.
Yes.
Actually, the major fatty acid, actually, the major fatty acid is 16 and 18. Whether there are actually there are few papers which is the preprint papers. Actually, they mention about the fatty acid, particularly related to PUFA. They might in our study we didn't find the striking change of the PUFA. Maybe different cohort may have different results. We didn't find the dry drastic change. Only here we found the PUFA. Actually, the free fatty acid found the increase. With other PUFA like DHA, EPA, we didn't find striking changes.
You have one question for one more, because we just got one question here.
Yeah.
Okay, Gina Barbosa is asking, can the distinguishing metabolites be used for diagnostic purposes?
Means the metabolites can be used as a diagnostic tool.
But have you found any from your study?
Yeah. You mean from my study, have use the metabolism as a diagnostic tool? Is this your question? Sorry, I didn't catch it.
I mean, I mean that she most probably wants to know that from your study, if you have found any key metabolite that can be used as converted to a diagnostic tool to differentiate COVID positive.
I don't know whether it can be used as diagnostic tool or not. Although currently the data show because it's just compared to health controls, and so whether it's specific to COVID or not, we need more study to answer this question.
Yeah, makes sense. Thank you very much.
Thank you.
I think I'm going to hand over to Brad now for the lightning talks.
Thank you very much, Fana. And thank you for the great talk. We'll now kick off our series of lightning talks. And I will just share my screen. Thank you. So our list of speakers today, we'll start off with Heike Schwendel from Plant and Food Research in New Zealand. So Heike, I believe I have your slides.
Oh, yes. Thank you.
Just let me know when you want it to change. Thank you.
Is there a specific way how I let you know?
Just say next slide.
Okay, thank you. Sorry.
We're not very high tech.
Okay, so yes, my talk is about resilience in berries after mechanical shaking. Next slide, please. So Botrytis cinerea is a fungal pathogen which is present basically everywhere and can affect a variety of plants, and it causes a significant loss of crop spread rate. So the top picture, you can see what it does to grapes, and you just generally have this moldy water loss kind of picture, and it can take over the whole plant, and you see the formations of brown spots. So obviously the crop can be ruined very easily, and it appears in humid, warm environments. So there's not really much you can do about it to prevent it from infecting. However, grape growers, so the horticulture industry has been severely affected by this. And it has been observed that when grape vines are shaken, and the shaking is done for thinning for a completely different reason, to reduce the crop of grapes and get higher quality product for wine making. So it was observed that when the vines are shaking temporarily, mechanically, for a couple of seconds, that actually had an effect on botrytis infection. So the botrytis infection was seen much less in these grapes, up to 50% less when they're shaken. And if you shake the vines twice, you can see even a further decrease. Obviously, you're losing some crop and debris and plant material, but the shaking as a stress-inducing factor has some sort of an effect on botrytis. So we used GCMS and LCMS and the Martin-Toffen-S to figure out, to understand what does the shaking do to the grapevine, and then hopefully be able to explain how this can prevent, or why this has such an impact on why has it increased the disease resistance in the grapevines? So we measured 93 primary metabolites. We tentatively identified 91 secondary metabolites, and we used the MALDI just to see which effect of the grape, and we only analyzed the grape berries, where is the main areas where they're affected. Next slide, please. So in the primary metabolites, and I'm sorry for the slightly crowded slide. So we can see that in the primary metabolites, we can see that especially amino acids and also fatty acids were affected and were more increased. And so the shaking seems to affect the shikimate pathway by increasing aromatic amino acids. And aromatic amino acids like tryptophan, tyrosine, phenylalanine are precursors for secondary metabolites, and we can again see that we observed in the secondary metabolites that hydroxycinnamic acids and flavonols like quercetin and epicatechin and all these associated plant metabolites, they're all increased, and they are linking, or they're leading us to believe that the lignin content might have been increased. So we haven't confirmed this yet, and that causes a thicker cell wall, which we can also see in the MALDI. So we can see that with the MALDI technique, we can see that the main effect is in the skin, in the outer layer of the grapeberry, which seems to thicken and seems to become more resistant, which then, of course, would explain why we see less botrytis infection, because the fungi just doesn't have that big of an effect of being able to get into the grapeberry. So this was a first trial. We have a follow-up study where we took more samples, more replicates, more sampling points, and despite COVID, we nearly achieved the more sampling points. And we will have a follow-up one where we get this even more detail and hopefully can really say how shaking can, that short stress of shaking can actually increase pest resistance, because if that would be a logical connection, that could actually be applied to other plants then as well. Next slide, please. There we go, my 5-minute talk. Thank you very much.
Wonderful. Thank you for that talk. And like I said, the chat is, the Q&A panel is where you can put your questions for Heike, and we'll come back to them at the end in a group Q&A. Next, we have Doreen Loh from Nanyang Technological University, Singapore. And I'm sharing, you're sharing your screen for this one, apparently. Doreen,
Yeah.
do you want to take over, share your screen?
Sure. Okay, great. Can you see my screen?
Yes, you're good to go.
Okay, perfect. Great. So thank you, Brad. I'm very happy today to be able to participate in the virtual podium, and I'd like to share some of my findings. That it's actually a continuation from my previous Agri Skills Fellowship in Rennes, France. So this work is a collaborative effort between 19 metabolomics platforms. And it was conducted in the European Cost Positive Action, where we evaluated the performance of PredRed to predict the retention times of 467 plant food bioactive compounds and their metabolites, involving 24 reversed phased LC chromatographic systems, and each of the systems contained entry data sets of 29 to 103 compounds that are chemically diverse. As you can see here, they have log P values ranging from -3.5 to about 10, and monoisotopic masses ranging from about 95 to over 900 Daltons. The motivation for this work was actually to contribute towards the annotation of dark matter in metabolomics, where there still is a very large fraction of molecular signals that remain undetected, especially with untargeted analysis. And part of this dark matter corresponds to the food metabolome, comprising of known food compounds such as polyphenols, glucosinates, alkaloids, etc., including phase 1, phase 2, and also gut microbial-derived metabolites that are recently receiving a lot of widespread interest due to their protective health effects, as well as their usefulness as food intake biomarkers. So these data sets, they were entered into PredRed, which is an open access database of compound retention times that was originally developed by Jan Stanstrup from the University of Copenhagen. PredRed then builds generalized additive models between one system and other systems in a database and generates a list of predicted retention times that have not been previously experimentally measured in our own systems. So we can then use this information as an orthogonal parameter for identifying other unknown compounds in our own systems. And in this work, we identified or we obtained more than 6,000 new retention and predictions all across the 24 systems. And here we can see that the predictions are highly variable, ranging from 91 to 700 over predictions over each of the systems, with a mean prediction error of 0.03 to 0.8 minutes, depending on the length of each system's LC runtime. And as an example here, in one of the chromatographic systems, we have more than 80% prediction of the entry data set. The figures is a little bit small, but the blue circles here refer to experimental retention time that was measured in our own system and then entered into the PredRed database, whereas the black circles represents the predicted retention time. And for some, you can also see a prediction interval. So in summary, the accuracy of the retention time depends on several factors, including the size of the retention data set that we enter into PredRed, as well as the size and also the number of common compounds that are shared amongst the systems in a database, and the type and the shape of the LC gradient of each our method. So to put it quite simply, the more data sets we add into the PredRed database, the more predictions we will achieve, including predictions for rare compounds and also a higher prediction accuracy. So overall, within the platform, we feel that PredRed really enables the scientific community to benefit from sharing retention time data. Most laboratories tend to have their own routine, semi-targeted or untargeted methods that covers plant food compounds in different matrices. So quite importantly in this aspect, PredRed is very user-friendly because it enables us to transpose retention times from one system to another system that differs in for example, mobile phase composition, the gradient, the flow rate, and also column dimensions. So yeah, please feel free to try using PredRed, and you can always email me or Jan if you have any questions. Yeah, thank you for your attention.
You get a very short bell. Thank you very much for that, Doreen. I will just resume the screen sharing, and we will move quickly on to our third lightning talk. And our third lightning talk is being presented by Sophia Lukinga from the Florey Institute here in Melbourne. And with that, I'm not sharing, of course. Oh no.
I think you are.
I am. Excellent. Away you go. Thank you.
Yeah. Awesome. Thank you very much. I'm just, I've got all my my data up there, but I'll take you through everything slowly. First, I'm going to talk to you about amyotrophic lateral sclerosis, which is a severe neurodegenerative disorder characterized by upper and lower motor neuron loss. It presents with muscle spasms, muscle weakness, and eventually patients die between 3 to 5 years after diagnosis. About 90% of all cases are sporadic without a known cause. But one feature that all cases of ALS kind of have in common, so both the sporadic and also the familial cases, is that they have abnormal metabolism. And a lot of patients are also seen to have hyperlipidemia, and it's like thought that that is an increased risk factor for like worsened disease outcomes. So quite recently, there's been more and more focus on this lipid dysfunction and specifically glycosphingolipid metabolism has been shown to be impaired. At the moment, the most common mouse model where we have modeled this is the SORT1 mouse model. However, only 3% of all ALS cases harbor this mutation. So in my studies, I've used a mouse model which represent the wider range of patients and like about 97% of patients, including some familial cases, but not SORT1 cases, they display TDP pathology. So here we use the TDP43 mouse model, and we treated these mice chronically with an already FDA-approved drug called Ambroxol, which is a selective glucocerebrosidase A2 inhibitor. And we aim to restore glycosphingolipid balance in the muscle and spinal cord. After 5 months of chronic treatment, we took the muscle and the spinal cord and we performed targeted and untargeted lipidomics in collaboration with Thusi, who many of you will know. In the first graph, I show you a little bit of behavior data showing that in the white bars, that is the wild type mice, the black bars are the TDP transgenic mice, and then in red you can see that the phenotype of the TDP43 mice is rescued, as in the mice perform better when they have been treated with Ambroxol. So when we did our lipidomics, so in yellow we see like yellow outlined is muscle and blue outlined is the spinal cord data. So first we did a t-test to see if there's actually a difference between the two genotypes. So we see that there's like on the score plot, there's a clear dissociation between the genotypes, indicating that there is profound lipid dysregulation. And we also see in our heat maps the top 50 dysregulated lipids, and there's a clear dissociation between the two genotypes. So then when we look at the interaction with Ambroxol, we see that Ambroxol mainly has an effect actually in the lumbar spinal cord. So that is, if you look at this panel number 4, that's the lumbar spinal cord data. So Ambroxol has more of an effect there, which is not surprising because lipid content is very high in the spinal cord compared to the muscle. So it's pretty. It was expected to be high and to have a higher effect in the spinal cord. So what we can conclude from this is that we have found a profound lipid dysregulation in ALS mice, and improving the glycosphingolipid pathway restores motor functions, and we can potentially use these highly dysregulated lipids as a potential blood biomarker. Thank you.
Thank you very much. Okay, wonderful. Now we have Josie Adamansky. Maybe I'll let you introduce the name of your institute, but I believe you're going to be sharing your screen with us. So feel free to take over. And we can see your screen, and we just need to be able to hear you. Still on mute from what I can see.
Hey, hi everybody. Now it's better. So I'm sharing this screen, which is showing me a lot of stuff I don't want to show, but do you see only my presentation?
We can only see your slides from my screen.
Oh, this is great. So I'm talking about the lipidomic analysis in human adipocytes today. I'm happy to join you. Very good morning from Munich. The sun is not set up yet. It's pitch dark outside. I'm talking about the lipids, lipid changes, which are observed during obesity. The best way would be to start for us to make the experiment in humans, but we can't. And this is because it is impossible to make a representative study on mechanistic basis if you're using human samples. So we switch to the human cell line, which is well established in the field. And the cell line has to be differentiated to perform the change from fibroblast to human adipocytes. We performed the analysis with SciEx technology using lipidizer. Out of 1,060 lipids, which were theoretically possible to be seen after QC, we have seen 743 lipids from 11 different classes. Now, the setup, first we have started the seeding of cell line and differentiated it with mixture of cocktails, performing maturation, and you can clearly see that you have accumulation of lipid drops. So you could make a staining with of lipids and see the colors coming up, but we have performed other analysis as well to verify the differentiation to adipocytes. For example, we have been following transcription of different factors, and they are following the known patterns for PPR gamma and CVPA. So we are sure that we're dealing with adipocytes. Now, the analysis showed that from the day 1 or 0, the seed out, to the day 20, where the adipocytes are fully differentiated, we have a continuous change in the representation of chain length and different classes in the lipids. Shortly speaking, from very diverse representation, we're actually having a mono representation of triacylglycerols. If you can focus on this panel, you can see that there are different changes showing that some lipids are dropping and some lipids are going up. And this is very specific description, which I don't have time to show up today. We performed not only the analysis of different lipid classes and individual lipids, but as well the correlation between different classes. So as you can see here, there are some changes which are anti-correlated and positively correlated. So some lipids are going up and some lipids are going down at the same time. And we have observed these differences between different classes. We have been looking at the classes and chain length. And the summary of outcome is that our cell culture model is representing a very good tool. It has been used successfully to discover new paths of lipid remodeling. I don't have time to show you all data. We found out without any big surprise that there is a class of more frequent lipids which are being stored, which are depicted here. And the message out of the correlation analysis is giving actually some new insight into the way the triacylglycerols are being synthesized. So we have, for example, phosphatic ethanolamines, phosphatic chlorines going into triacylglycerols, and ceramides and going into sphingomyelins. So these are actually the descriptions how the remodeling of lipids is going on during adipogenesis. Thank you for your attention. This manuscript has been part of our PhD work of Florian Miller, and the authors are depicted on the slides. And the manuscript, full details, was just published in a great journal. I hope Peter is online in the Metabolite just this year, describing all data there. Thank you very much.
Thank you very much. That's excellent, excellent presentation. We are now going to go with lightning talk number 5, and this is another one where I have to relinquish control of my screen, but Luo Shi Hui is going to give us a 5-minute lightning talk. Luo Shi, do you want to steal the control from me? And you will need to unmute yourself as well.
Sorry.
That's
can you
okay?
hear me?
Yes,
okay.
we can.
Okay. Have you seen my slide?
We can now, yes.
Is it?
It's not in slideshow. Yep, now it's in
Okay.
full slideshow.
Okay.
Take it away, Luo Shi.
Okay, so can I start now? Okay. Is it?
We're now on the other monitor, the other screen, or the other
Another monitor?
workspace. That one. That's the one.
This one? Is it?
Yep. Good.
Okay. Sorry. So good morning, good afternoon, everyone. And I'm Shi Hui, and currently year 2 PhD student in Kaohsiung Medical University, and my professor is Ko Liang Ping. So today my topic will talk about the dual function of bioenzyme X for extracorporeal detoxification of the atherogenic lipoprotein. So this is our goal. We aim to develop a extracorporeal device which can remove the atherogenic lipids such as LPC and ceramide in the circulation. Okay, so nowadays the cardiovascular disease remains the leading cause of death globally, but still have some controversial issue. So now the current strategy against cardiovascular will mainly focus on how to lowering the level of LDL. But here will form another issue. So we try to lowering the LDL. Is it can preclude the ASCVD? So from the results we know cannot, because although the patient control their LDL under 100 milligram per deciliter, they still have around 45% of the risk to have atherosclerosis. And the other issue is if try to lower the LDL, so intensive dose of statin will increase the side effect, and mostly the patient will stop taking the statin due to the side effect. So in our lab, we divided the LDL into 5 subfractions from L1 to L5, and we found that only the L5 subfraction is toxicity. So maybe L5 LDL is the culprit of cardiovascular disease. So in various diseases, we can find that the L5 LDL was extremely increased, especially in the STEMI patient. Their L5 will 15-fold higher than control. So we checked the toxicity of the L5 LDL, and we found that there are 2 atherogenic lipids like LPC and ceramide may contribute the cytotoxic effect of L5 LDL. So we further set up a study design. Then we found an enzyme which can hydrolyze LPC and ceramide at the same time. So we're setting up the stable clone of the enzyme, and the sequence was checked by single sequencing, and the results also checked by the Western blot. And we purify the enzyme by immunoprecipitation and also HPLC system. So then finally we check the enzyme function with LC-MS system. So we incubate the enzyme with lipid standard and also the LDL from patients and high-fat diet pigs. And in the end, we also coat the enzyme onto the resin and we check the function. So our results showing that our enzyme can hydrolyze around 71% of LPC and 90% of ceramide. So our conclusion is we aim to develop a bioenzyme extracorporeal device So we hope that it can be a novel therapeutic approach to the cardiovascular disease in the future. So thank you for your attention.
Thank you. Thank you very much. Excellent. All right. Once again, I'll remind our attendees that the Q&A panel is available for any questions. But right now, we can move to our last lightning talk before our closing keynote. And I will just jump back into Sivamoke Disuk from Graduate School of Engineering in Osaka is going to talk to us today. You're still on mute.
But Siomok need to share, yes.
Oh, okay.
Last minute, yeah.
Please take over control.
I will share the screen.
No problem. Looking good.
Right.
Looks good.
All right. Okay. Okay. Thank you for having me. So my study is about the yeast in Yarovia genus. So for the long time, the yeast in this genus was only identified by only one species, the Yarovia lipolytica. However, like 10 years ago, when the phylogenetic analysis of the DNA sequence available, more strains have been included in this subspecies and this genus. So what is interesting about this genus is that it's a very versatile for the carbon source consumption, and it could use to produce the various type of industrial importance commodities like the functional fatty acids or biofuel, something like that. And it's capable of producing this and store lipids in like 20 to 20% of its dry cell weight. Now, one of the most important, commonly known thing about the yeast and the lipid production is when we reduce the nitrogen content in the medium, it tends to accumulate the lipid more. However, no one have elucidated what happened with this newly identified Yarovia species. That's what I do now. So in our experiment workflow, we took several Yarovia lipolytica strains from Japan collection of microorganisms, and then we varied the carbon to nitrogen ratio. So the first one is the standard condition, and another one is the nitrogen limiting conditions. And then we sample in the 36 hour. Well, this was optimized with the previous experiments. And then we extract the polar phase using the liquid extraction. Now we want to analyze the primary metabolites. Then we use the LCMS, the iron pair reverse phase, LC-QMS, to analyze the primary metabolites. So what we found, this is the PCA analysis of the 6 strains of Yarovia species. We found that, of course, in normal condition, because they are different species, they have a different metabolome profile. However, we found that they respond to nitrogen limiting in a similar way. However, when we analyze the nitrogen limiting condition alone, we found that even in the nitrogen limiting condition that we see that they respond in the similar way, the response is a little bit different for each strain, and it can be captured by the metabolome profile. So next is what we found. What is the core similarity between these strains are that when we reduce the nitrogen content of the medium, the purine and pyrimidine metabolism are most affected. Notably, the ATP, ADP, AMP, and the GTP, GDP, GMP on the purine metabolism was similar in all strains. You can see, sorry, maybe it's a little bit small, but these metabolites have the very same profile. However, the other profile of the purine and pyrimidine metabolism will be different, varying in each strains. Also, in pyrimidine metabolism, the thing that is same in all strain is the UTP, UDP, and UMP. So, in summary, this metabolomic analysis revealed that the Yarovia is a very diverse genus, even though the phylogenetic analysis say otherwise. Although the response in nitrogen limiting condition is similar, however, they have each strain have their unique response, and based on results, when we doing some trait optimization, we should consider this kind of changes. And the take-home message is like, maybe we demonstrate that the metabolomics is a powerful tool for this purpose. Okay, thank you for your listening.
Thank you. Okay, so that concludes the last of our lightning talks. We're now opening up the Q&A, so if anybody has any questions, and we certainly have a couple. I guess my first question, if I can selfishly jump in because I have the microphone, is for Heike. With the botrytis on the grapes, I know that the physical shaking causes a change on the skin. That process is for thinning out some of the grapes. Is there increased airflow or anything that could explain the reduction in botrytis, or is it all due to the skin hardening and the changes in the skin?
Yes, so there is the possibility that that is the increased airflow, the removal of already slightly infected berries, or we definitely consider the physical aspects as well. And this is what I presented is really just a very, very small part of a much, much larger project where we investigate botrytis. And Farhana, I could probably tell you a bit more detail about it. But yes, this is we consider that the physical part definitely has an effect, but also metabolomics was part of this trial, and the changes we can see within the metabolomics, and also what other studies have reported when they studied the effect of wind or other mechanical shaking on the grasses and how it affects disease resistance, also following the metabolomic pathway. There's certainly a metabolomic aspect to it. Whether it's exclusively, it's another question, but yes.
Cool. And we have a question for Doreen in the chat, and I'll also remind people while I read this one out that after the final keynote, which will be delivered shortly, there will be an open discussion after the keynote question and answer. So still time to get those questions in. Doreen, I'm going to read this verbatim, but it says, PredRet looks cool. Can it be used to predict retention time of lipids in different LC systems?
Yeah, absolutely. You can use it for all sorts of compounds, food compounds, bioactive compounds, lipids, as well as also, I think there's some pharmaceutical compounds in the system as well. So yeah.
Okay, cool. I think that has been answered. And do we have any other questions from the floor or the panelists? I will take that as a no at this stage. And with that, I will hand it back to...
I would have a question about the
Sure.
shaking as well. Isn't that, Heike, did you say that the Shikimate pathway is going up?
Yes, we can see more. I mean, it's affected, so we can see more of the aromatic amino acids. Yes.
Like I remember, I think from bacteria, from Corynebacterium, that under stress, the Shikimate pathway always goes up. No. So
yes.
Is that shaking just like a general stress response? So they're just more on their toes to.
I mean, this was just a really small trial to start. We now have a second-year trial where we have more sample data and so on and so on. But the theory is that we have this short burst of stress which doesn't damage the plant in any way. It's just this short stress burst without any long-term effects. But that changes the plant, or in our case, the grapes, so much that the stress response actually prohibits future stress responses, or the effect of future stress responses as the infection with a pathogen. So that is the theory. And if this would be true, then mechanical shaking could be used in other plants, or this short stressor of the plant could be used in other plants to prevent susceptibility to other pathogens, or potentially even droughts or flooding events or whatever, because the plant has already gone through that process and recovered. Kind of like sounds really crazy, but like an immunization process in a way. So yeah, that's our theory.
Thanks.
Okay, wonderful. Fana, I'll hand it over to you to introduce our final keynote speaker. So take it away, Fana.
Well, thank you so much, Brad. Yes, welcome back, everyone. I mean, you have been here. But what I was going to say, that it's my pleasure again to introduce Shastia Putri. She's an assistant professor of Osaka University, and she also involved with the Institute of Technology Bantung, Indonesia. And I actually know Shastia for a while, since she was the first chair of Early Career Member Scientist Network of Metabolomics Society, and she also played really instrumental role in Metabolomics Society as a board of director from 2015 to 2019. So Shasta is going to tell us more about her work, how she has been using mass spectrometry-based metabolomics for quality improvement of high-value tropical byproducts. Shastia, I'm just handing it over to you. And thank you for coming here.
Yes, thank you so much, Farhana, for your kind introduction. So may I start sharing my screen?
You may. Take it away.
Yes. Okay. So first of all, I would like to really thank all of the organizers for making this happen. I think it's really great that we have an event that connects metabolomics researchers in the Asia Pacific region. I think it's about time, and I'm really excited to be here today. So today I would like to talk about several examples of my research on the quality improvement of high-value tropical bioproducts. So where I come from Indonesia, it's a very rich country, rich of bioresource and abundant food production, but crop loss often occurs. So this motivates me to do something about the application of metabolomics for tackling these issues. So most of the main issues resulting in major crop loss in my home country is either caused by disease, poor post-harvest management, or just simple low quality. So how metabolomics can tackle these issues, and we can of course study about the plant diseases and investigate the cause of diseases. We can also aid systematic and effective post-harvest management, and I would like to talk about today mostly about the quality improvement. So we all know that the application of metabolomics is really wide, and we have excellent speakers today in the session and also in the other series of the virtual podium, talking about the many different facets of applications of metabolomics. And just last month, I talked about some of my research on the bioindustry application. And I'm really excited to show you the latest research that I have on coffee metabolomics. So these research topics are done in collaboration with many of the research institutes in the Asia Pacific region. And I hope that this would be a good chance also for us to start new collaborations through this virtual podium. So the concept of food metabolomics is pretty simple. So usually we should have a food function, and then we can analyze the components, and then using statistical relation, we could analyze the relation between these food functions and the components. So many different ways that we could apply metabolomics research. It can be either discriminative, informative, or predictive. So I would like to give an example of each and every one of them. So the first topic that I would like to talk about is one of my favorite. It's coffee. I think everyone here probably needs coffee at around this time. In Japan, it's already almost coffee time. So I would like to talk about Indonesian specialty coffee. So you may know that there are many different grades of coffee. So the specialty coffee is gaining a lot of attention in recent years, and is always usually most commonly sold as single origin coffee. So it has to have more than 80 points from a total of 100 maximums. It comprises of 10 attributes as shown here. And the most commonly used like gold standard to determine the quality of specialty coffee is using the Specialty Coffee Association of America, or called ACAA cupping test. So there are many different research on coffee. So various factors affect the quality of coffee, many different kind of component profile analysis, let it be volatiles or non-volatiles, and also description on the coffee quality. So very few research have dealt with the correlation between the coffee quality and the component profile. And actually, this is very useful to later on give feedback to improve the quality of coffee, and also to develop more robust quality assessment. So the objective of this study is to propose the quality marker of specialty coffee by regression model-based metabolome analysis. We analyze various kinds of specialty coffee, subject that to cupping test, and then apply this to the component profiling. So there are 10 kinds of specialty coffee that were evaluated based on the ACA protocol by a skilled panelist. This is a licensed panelist, Q grader, from the Indonesian Coffee and Cocoa Research Institute. And the range of the coffee score ranges from 80 to 86. And we performed GCMS analysis to analyze hydrophilic low molecular weight compounds in coffee extracts, and then subject that to an orthogonal projection to latent structure, also called OPLS, to find the relationship between Y and X variables. For the explanatory variables, we used the metabolome profile, whereas the cupping test score was used as a response variable in this model. So here there are two terms that I would like to introduce. It's variable influence on projection, or the VIP score, and the regression coefficient. It can be positively contributed to the model or negatively contribute to the model. So here it's very interesting to see that for each of the model that we create, the top VIP compound showed that galactinol was proposed as a metabolite with high VIP. So in our analysis, galactinol was proposed as a quality marker based on the prediction models of 7 attributes and the final score. So this is for the first time we propose a high correlation between galactinol and flavor of specialty coffee. And we also measure the quantitative analysis of this galactinol, and it is indeed higher compared to when we compare the highest and the lowest specialty coffee based on the score. And we performed also whether the panelists can perceive galactinol when we added the galactinol into coffee. But the contribution of galactinol to coffee flavor could not be confirmed by human sensory tests, most probably because the quantity is very, very low. So from this study, we would like to propose galactinol as a quality marker of specialty coffee, and further research to validate this is presently ongoing. So I would like to continue also on several examples that I have for coffee, still coffee story. And other than to propose the quality marker for coffee, we can also do this kind of metabolomics research for authentication purpose. Here I would like to show the example using Indonesia's Kopi Luwak, which is the world's most expensive coffee that is digested by civet. It's a kind of animal. It's kind of a cat. It's an animal that is a kind of cat. And here, using OPLSDA, we could separate coffee that is digested by luwak and not digested by luwak. But if we employ PCA, it's very difficult to separate because the coffee variability based on the species or region will give the bigger influence to separate the coffee. So we were able to separate the two classes using discriminant analysis. So this was one of the first examples of coffee research for authentication in 2013. And then based on the S-plot on OPLSDA, we subjected the candidate markers and then subjected to validation of the applicability of the discriminant marker. And we proposed several markers for determining the authenticity of kopi luwak. So this was the first study on the establishment of an authentication method for world's expensive coffee. And we also did a biological experiment where we fed the kopi luwak. We fed the coffee fruit to luwak, and then we measured the before and after, and we validated that indeed the markers were increased after digestion. So this research was very well perceived, and it was one of the gateway that opened a lot of possibilities for me to start a lot of different research on the application of metabolomics for various kinds of tropical bioproducts. So just last year, I also completed the mapping of Indonesian specialty coffee based on different regions, and it was published in Metabolomics. And we also had several examples on how to predict the ripening stage of fruit. For example, we could use metabolomics to evaluate different post-harvest treatment in mangosteen. So this is one of the most important fruit in the Southeast Asian regions. It's also called the queen of fruits. So it's also one of the most exported fruit from Indonesia, but it ripens really quickly. So very few methods are effective to actually prolong the shelf life. By only doing the storage in low temperature, mangosteen shelf life can be prolonged. So we compared what happened between the mangosteen that ripens in the normal condition in room temperature versus the mangosteen that ripens in which ripening is inhibited in low temperature. And we found that the increase of some of the metabolites associated with ripening was indeed inhibited in the storage in under room temperature. And we also can observe that there are different kinds of metabolites that were significantly changed over time throughout the course of ripening, and those were galactose and galacturonic acid. But it's interesting to note that no significant changes were observed in the flesh part. So previously, the result that I showed you was on the peel part. So the score plot showed here that there's no trend for delaying mangosteen ripening process in low temperature treatment. So although we could see there are some differences between the two treatments, but I think we still have to find effective ways to prolong mangosteen shelf life, not only looking at the outside, but also looking at the flesh part. So similar studies have also been applied to a different kind of fruit. I explained about mangosteen, which is a climacteric fruit. We also recently published this year in Metabolites for the ripening of pineapple. And I would like to close this talk by mentioning that there are many different ways that metabolomics can be used for food science and technology. For example, for discrimination, for informative evaluation, and also for prediction of important quantitative phenotypes that are very useful in the food science and technology. Hopefully, this will also help us to improve the quality of our produce and also apply that for the food industry. So I would like to thank everyone for your kind attention. And I think application of metabolomics can be a very powerful tool for strengthening a nation's food security as well. Thank you very much.
Thank you, Shastia. That was an excellent presentation. Actually, I have to admit that I'm a fan of kopi luwak. Not that I can buy it all the time, but it's really good. I just have one question because your area is quite similar to the kind of work I do. I'm mostly working in wine and other fruits we find in New Zealand. So you said that galactinol was a marker for coffee quality, and you have associated that with the flavor. Do you think, because in when we do study for wine, we find that sugar alcohols are more related to mouthfeel? So do you have any indication that galactinol could be related to the mouthfeel, and this is why most probably the coffee panelists are picking it up or your study is picking it up as a marker for quality?
Yeah, it's a very interesting discussion because we haven't actually found any studies relating galactinol with the coffee quality. So it's a very new finding. But when we try to do the quantitative analysis, it's present in very, very, very small amount. So I think it's probably not the panelists that we use for testing that were involved in the testing, were not able to perceive galactinol differences under such low concentration. So there's a possibility that we could increase the concentration. So we try to do that by increasing it 10 times or 20 times more than the endogenous concentration. And they could see that there's a difference, and it becomes sweeter, and also, like you say, mouthfeel, or there's a different kind of flavor and body that changed. But we cannot still conclusively conclude like that it is related to what kind of attribute, because the panelists still cannot discriminate with the galactinol addition alone. So maybe it has to work with other compounds. It's quite difficult to conclude.
Is there any other question for Shastia?
Question.
I think everybody has a question.
Okay, I jump first. Saskia, this was really, really nice. Is it mainly based on the species of the coffee, or the treatment and the roasting and the storage, and like how many days after roasting and all of that?
Okay, so in the paper that I published on the different kinds of specialty coffee from all over Indonesia, we compared 3 variables at once, which is a roasting process, Arabica versus Robusta, and also different region. And in PC1, roasting process is the most influential factor. So in PC2, it comes species. So we cannot see any distinction between the regions. So we have to analyze the samples independently. So only green Arabica and green Robusta, then we can observe the geographical origin. So yeah, that is the order of how it's more influential. So the roasting process is definitely the most influential process in changing the coffee metabolite profile more than the species. I hope this answers your question.
Yes, thank you.
Okay, great.
No one is asking. Yeah, Brad, you want to jump in?
Because I can go next. Yeah.
Another question. Yeah. Okay.
I certainly know about Luwak. I haven't had very much of that coffee, but are we closer to understanding how to generate that coffee without the obviously involvement of animals? Is that something that's ongoing? Sorry if it's obvious or it's been done so many times.
Yes, it's a really, really good question. And it actually, I only have 15 minutes, so I couldn't explain everything. But I already have a patent on making kopi luwak without the animal, because it's related to the animal issues. Like we don't want to make a product that use animals, because actually kopi luwak is naturally produced in the wild. So the civet in the wild forest are free to roam and then choose the best coffee cherries. And that is also one of the selection how kopi luwak has a very, very distinct flavor, because of the ability of the animal to select the best coffee cherry. Yeah. But once you put them in the cage, it puts them under stress. So actually, and then we select the coffee cherries for them. So I think the quality of the wild kopi luwak and the not wild kopi luwak will be different. So we isolated the bacteria from the feces of the excrement of the luwak, and then we combined many different kinds of starters, and then tested against our metabolome profile to see which one has more similarity, or the one that could increase the flavor. And then it cannot make the coffee like Kopi Luwak, but it can increase the quality of coffee based on the cupping score. And we already published that in Metabolomics Journal. It was published earlier this year in January. So if you're interested.
Cool.
So it's possible to increase the quality of coffee through controlled fermentation.
Excellent.
Yeah, I believe that is more going to the digestion aspect of it, because when the animal's gut, there is a anaerobic kind of digestion fermentation happening. So most probably we need to do more studies in terms of understanding what is happening there.
Yes, and it's also affected by what the luwak is eating other than coffee cherry sometimes. If it's a wild process, that also affects the microbiome in the luwak gut. So maybe it's not possible to make it exactly like Kopi Luwak, but at least it could somewhat increase the coffee cupping score significantly.
And I'm assuming reduce the cost as well, right? If we can do it in an industrial fashion.
It's true. You can increase and upscale the system too.
So we are actually running out of time. I apologize for that, and I would like to hand over to Brad. And yeah, Brad, here you go.
Sorry, I'm actually responsible for us going over time there, so it's all my fault. Thank you very much. Fascinating talk, and I'll probably will look up that paper you mentioned as well. So thank you again to all of our speakers, and we have all of our lightning talk speakers are still with us. So if there is any other questions or comments or anything, now is the time to bring those up. And so we have a few more minutes, but yeah, I think given previous weeks, we've certainly hung around for a little beyond the official close time. So yeah, any? I think there was a question, but it disappeared, or was it answered? We might have answered it. Thank you, Doreen, for answering that one. Cool. Okay. Well, look, thank you to our panelists and to both of our keynote speakers, as well as the lightning talks. It's certainly no mean feat to squeeze information into that format. I really do enjoy those talks. For everybody who's attending, I believe in recent weeks, if we've got questions via email afterwards, we've sent them to the various speakers so that they could respond directly. Yeah, I don't know if Fana or Anna or Tusi, if you have any closing comments, apart from make sure you join us next week for the final installment. I'm not sure whether I'll be timekeeper or chair next week. I'll have to fight it out with everybody for my gong abilities.
Thanks,
No problem.
This agenda. Next slide, if you go to the
Thank you,
next slide.
Tusi. Thank you very much for showing me that. Yes.
Yeah.
got a couple of talk titles that are sort of yet to come in. But yes, it looks like we've got a solid lineup, and definitely everybody's going to definitely dial in for Peter Meikle, who joined us today. I did notice he was with us, who will be closing out the virtual podium for Asia Pacific next Friday. Same Metabolomics channel, same Metabolomics place. All right. With that, thank you all for attending.
Thank you, everyone.
Thank you.
Have a great weekend.
Thank you so much.
Stay safe.
Thank you for having me.
Thank you. It's great.
Everyone.